Thank you, Per. Our last talk is bridging brain coordinates and machine learning for surgical targeting and morphometric mapping. And that's from Alaa Taha of the University of Western Ontario, London, in Ontario, from the Biomedical Engineering Department. Welcome, Alaa Taha.
Great. I have to admit, being part of this session is actually quite humbling. But what I hope to do today is take you through how what started as a local brain hack project at our own institution sort of transformed into more or less of a program to annotate the human brain or annotate landmarks in the human brain. And this framework has made its way sort of to quite a number of applications in human brain mapping and clinical applications. And so I want to take a moment to acknowledge the support of my supervisors, Dr. Jonathan Lau and Dr. Ali Khan, and many of the lab mates and collaborators who made this work possible. So I want to start with some of the clinical insight or motivation behind this work. Deep brain stimulation is a neurosurgical procedure that involves the modulation of brain activity by electrodes. It has been studied for a wide spectrum of neurological diseases. And with more than 150,000 cases implanted worldwide, we know that it works quite well for managing various neurological diseases, but also including Parkinson's disease, and that's what you see on the top right-hand side of my slide. What I want to emphasize is that millimeters matter in DBS. Deviations on the order of 2 millimeters can be the difference between optimal therapeutic benefit and side effects. And a common question that's relatively asked in the DBS literature is, how is this one DBS electrode modulating the large brain circuits that are implicated in, say, diseases like Parkinson's? And so I want to review the methodological aspects of DBS and registration. And typically, patients come to the clinic and they acquire a wide variety of multimodal MRI data, and we register all this data together to a safety scan called the Gadolinium enhanced scan. And we can also employ a brain atlas into the mix, and that provides sufficient contrast and information to plan the trajectory of 2 electrodes. The neurosurgeon implants these electrodes and quantifies the error of their implantation. But the neuroimaging researcher is interested in pooling this and studying this as a group or in a population. And so we employ a nonlinear registration to a brain atlas. And the output of that would look something like this, where you'd have, in this case, 25 electrodes from 25 different patients visualized in a common space, targeting a structure like the SDN or the subthalamic nucleus shown here in orange. What I want to emphasize is many of these registrations are typically only qualitatively reviewed. And so when we look at the SDN, which spans about 11 millimeters in maximal histological dimension, and look at the point cloud of the electrodes or electrode contacts, a good question to ask is how much of this variability that we see is biased or influenced by registration. And so to approach this problem, we need more sensitive registration metrics, and that's where our work with the anatomical fiducial framework comes into play. Over the past 5 years, we've validated 32 coordinates that can be placed within an accuracy that's less than 2 millimeters if you follow the protocol that we've published online. We've also released quite a number of data sets with annotations. And the idea behind the AFIDS framework is quite simple. We have 2 brains that we want to morph together. We look at the vector between the ground truth coordinate and the homologous coordinate on the moving scan. And that vector has a magnitude that communicated in millimeters representing the error, and the direction, which communicates also the direction of that error. And we've shown in a number of studies that this anatomical fiducial registration error derived from our AFIDS actually provides a much larger dynamic range relative to the commonly employed voxel overlap metrics. And we've shown that, as you see on the y-axis, for example here, the AFRE, which is anatomical fiducial registration error, you get a dynamic range ranging from 0 to 10 relative to the more binned, unitless dice overlap. And so the AFIDS framework is quite sensitive to errors in the millimetric scale, but it does require manual annotation. I want to take a couple of slides to show what can be done with our data. So we've released 5 data sets with ranging magnetic field strength, surveying various healthy and neurodegenerative disease, constituting more than 5,000 landmarks. Here's a heat map showing all the 173 subjects. And what we did is we registered all of these subjects to MNI space using fMRI prep. And that heat map represents the errors. We can investigate this error more locally at the different anatomical structures or landmarks, or even look at this error more globally, tallying across all landmarks in patients. And typically, fMRI prep seems to have performed with an accuracy of 1.6 millimeters. What's even more interesting to me is when we start looking at this error across the different data sets and mapping where they come from. We did a PCA on the errors of the different data sets, and what came out was interesting clusters that map back to some of the individual field strength or data sets that come from. And this is very relevant for studies looking at various populations in a common space. We also can do various comparisons with various registration algorithms and software in the literature, and we did a quick one here. There's certainly more to this story, but it's more of a one-slider about what we can do with this data. And so this motivated the use of our data to create what would be the AutoAPHIDS project, which is an automatic landmark regression framework. The APHIDS project, since its inception, has been grounded by open principles, and so we design, and so AutoAPHIDS is no different. What I want to emphasize is AutoAPHIDS is not simply a bunch of trained models to perform landmark regression. It's also a framework that you can use to train your own on your own data. And so we release models and the framework to do the training. And so here's how AutoAPHIDS performed on our data set with a global median of 1.28 millimeters, taking about 1 minute per scan when we parallelize that across all aphids. That actually puts us quite competitively with human raters doing the annotations themselves. We wanted to compare how AutoAPHIDS performs relative to other registration tools. Just a point I want to make here is, in this particular case, we moved the coordinates from MNI space to native space, allowing for a pairwise comparison across all patients. And what we observe is that AutoAPHIDS seems to perform a bit better than the 2 approaches that we tested here, within the range of 0 to 2.5 millimeters with a thinner tail in terms of outliers. What we found was interesting is the model seems to have generalized even on hippocampus laser ablation cases. We show here a laser ablation case with the ablation of the hippocampus highlighted, outlined in red, and the prediction for the model pointed in a red fiducial relative to the ground truth placement that we have as a reference in MNI. So it was quite interesting to see that generalizability, and there's certainly a lot more to tell here with regards to mapping lesions and ways to think about coordinates and clinical outcomes. And so really the framework that I've outlined so far has been focused on annotating data or annotating a wide variety of MRI scans. And we've used this data to create various approaches to recreate the same kind of data ultimately. And really the missing piece that I want to highlight in the next set of slides is the ability for us to extrapolate that to various clinical and human brain mapping applications. And so I want to bring things back to the DBS space, and what you see over here are coordinates in MNI. You see our typical aphids, 4 of them that are close to a surgical target called the STN, introduced earlier. And you see the electrode tips shown here in gray. And when we did a correlation between the errors of our aphids and the variability in electrode tip position, after doing a PCA, we found that the first principal component of errors explains about 28% of the first principal component of the electrode errors. And so really all that means is what we can start doing is thinking about the errors at aphids to ultimately correct some of the errors at electrodes. And that allows us to tighten up the electrode distribution and ultimately study DBS populations a bit better. Another application I want to highlight, and that's also the focus of one of our posters here, poster 006, is thinking about applying the AutoAPHIDS model in native space to predict other surgical targets. And so many of our anatomical fiducials or aphids circumscribe subcortical structures and surgical targets. And so we thought if we can build a model that can take these coordinates and predict the surgical targets, then perhaps that could allow us to circumvent registration altogether. And so we did that with the most common target called the subthalamic nucleus and performed with an accuracy that's just under 1 millimeter. But what was really interesting is this approach seems to have generalized across a variety of scans. So if you have a scan on which you can place the landmarks that we used to build the model, then now you become a bit more scan agnostic. What was also interesting is it also generalizes across other targets in close proximity, where we did a quick leave one aphid out analysis here, leaving one of our coordinates each time and repredicting it. We get a ballpark of about 1 millimeter of error. The last and final application I want to just highlight briefly today is thinking about these landmarks or coordinates as sort of a fingerprint for the brain. If you were to trace the pairwise distances across all the 32 coordinates, you would get a 496 distance vector that we can analyze in different ways. And so we applied our auto-aphids on an amalgamation of 2,000 scans from open data sets. And what we can start doing is thinking about changes across aging, with regards to important metrics like interventricular distance, or even clinically relevant metrics like the anterior and posterior commissure distance that we show here. So with that, I want to thank you all for listening, and I'm happy to take on any questions or discussions.
Thanks, that's a tremendous work. Any question from the audience? Yes,
just a naive question. I think you mentioned that the manual landmark you put on is like 2 millimeter error from the human, and then you compare metrics. So how, when you say this particular software has a 1.5 millimeter error in co-registration, how do you remove the manual?
Yeah, that's a great question. So the question is really, if the human rater can place these points with an accuracy that ranges between 0 to 2 millimeters, and this is actually the inter-rater accuracy. So if me and you place the scan, the inter-rater error would be 2 millimeters. And so if humans perform or place these coordinates with that accuracy, how can I claim that the model performs a bit better than that? And the answer to that is twofold. Well, we generate the ground truth coordinates from multiple raters. And so we take the average of multiple raters to achieve what we call is the ground truth. And so that's sort of how I can look at the prediction and see that whether it's better than the benchmark or not. And yeah, I think that pretty much sums up. I hope that answers the question.
Thank you. Any other question? If not, well, I mean, congrats again. I think it's such hard work, and hope that we'll go in eventually in clinical settings. I think that's.
Thank you. Appreciate it.
I would like to thank everyone, all the speakers, all the people who have actually coded all those tools and things, and you for your presence and your questions. And thank you again.